Weighing in at just 600 milligrams, the pituitary is the neuroendocrine command center of the human body. Its physiology is dictated by its physical location. It sits entirely encased within a rigid bony vault at the base of the skull known as the sella turcica. Because the gland is bounded by solid bone ventrally, expanding pituitary tumors cannot grow downward. They have exactly one path of least resistance, projecting straight upward. Right above the gland sits the optic chiasm. When a mass forces its way out of the sella turcica and compresses those intersecting optic nerves, the patient loses their peripheral vision, a clinical presentation known as bitemporal hemianopia. The gland is divided into two distinct lobes with different delivery systems. The posterior pituitary is supplied by the inferior hypophyseal arteries and receives direct neuronal innervation from the hypothalamus. This direct structural connection makes the hormones stored in the posterior lobe, specifically arginine vasopressin and oxytocin, highly vulnerable to physical trauma. A lesion on the pituitary stalk or the hypothalamus can instantly disrupt their release. The anterior pituitary operates on an entirely different mechanism. It lacks direct nerve connections, relying instead on a microvascular network called the hypothalamic pituitary portal plexus. This portal plexus allows the hypothalamus to send precise pulses of regulating peptides directly to the anterior endocrine cells. Because the blood does not enter systemic circulation first, these chemical signals arrive at the gland without being diluted. The rigid boundaries of the sella turcica define the neurological symptoms of tumors, while the precise vascular and neural wiring defines the biochemical vulnerabilities of the endocrine axes. Long before it sits in that bony vault, the anterior pituitary begins in the embryo as Rathke's pouch. Every endocrine cell in this lobe originates from a single pool of pluripotent precursor cells expressing the SOX2 gene. This genetic tree shows a SOX2 cell differentiating into 5 lineages via sequential transcription factors. Prop1 establishes 4 groups: growth hormone, prolactin, TSH, and gonadotropes. A Prop1 mutation destroys these pathways, causing combined hormone deficiency. The ACTH line remains unaffected, developing through the exclusive factor TPIT. A mutation in the TPIT gene yields an isolated ACTH deficiency. Without ACTH to stimulate the adrenal glands, the patient will present with secondary adrenal insufficiency, while the rest of their pituitary function remains intact. Further down the cascade, terminal factors lock in the final cell types. SF1 and DAX1 dictate the gonadotropes, while the factor PIT1 terminally controls the expression of growth hormone, prolactin, and TSH. Because these developmental pathways are fixed, identifying a specific transcription factor mutation allows direct prediction of the clinical deficiency syndrome the patient will face. Under the PIT1 genetic lineage, we find 2 main cell types: somatotropes, which make up 50% of the anterior pituitary, and lactotropes, which account for about 20%. These lineages both utilize the type 1 cytokine receptor family. Intracellularly, this means they both rely on the JAK-STAT signaling pathway to exert their effects. For the somatotropes, growth hormone synthesis and release is stimulated by GHRH from the hypothalamus, while somatostatin sets a constant basal inhibitory tone. Once released, growth hormone drives linear bone growth by prompting the liver to release IGF-1. It also acts as a systemic anti-insulin agent, making it inherently diabetogenic. We can target this receptor physiology clinically. The drug pegvisomant is a direct growth hormone receptor antagonist. By blocking the cytokine receptor, it shuts down the JAK-STAT pathway to treat acromegaly. Then we have the lactotropes, which secrete prolactin. Prolactin represents a unique exception to standard pituitary physiology. Unlike others, prolactin is under predominant inhibitory control. The hypothalamus sends dopamine down the stalk, binding D2 receptors on lactotropes to suppress prolactin. Severing this stalk cuts the dopamine supply. Without inhibition, lactotropes multiply and flood the bloodstream with prolactin, causing paradoxical hyperprolactinemia. In addition to dopamine's inhibition, prolactin is also driven by TRH, the same hypothalamic peptide that stimulates thyroid-stimulating hormone. Because of this inhibitory wiring, a structural mass compressing the stalk silences the release of growth hormone, ACTH, and the gonadotropes, while massively elevating prolactin. Next are the corticotropes. In primary adrenal failure, such as Addison's disease, cortisol levels drop. The pituitary responds to the loss of negative feedback by ramping up production, churning out massive amounts of the POMC precursor protein. As the cell cleaves all that excess POMC to make ACTH, it simultaneously generates enormous quantities of alpha MSH. Because alpha MSH stimulates skin pigmentation, this process causes the hyperpigmentation seen in Addison's disease. The corticotrope axis demonstrates how the cleavage of an upstream precursor protein directly dictates the observable clinical symptoms in a patient. The gonadotropes make up about 10% of the cells. Their activity is governed by GnRH, which is regulated by the upstream gene product, kisspeptin. The gonadotrope axis is highly sensitive to timing. To stimulate the release of luteinizing hormone and follicle-stimulating hormone, GnRH exposure must be pulsatile, arriving every 60 to 120 minutes. Continuous uninterrupted GnRH exposure causes profound receptor desensitization, shutting down LH and FSH release. This mechanism serves as the basis for long-acting GnRH agonist therapies used in prostate cancer. While testosterone and estrogen provide broad negative feedback, the peptide inhibin selectively suppresses only FSH, while activin independently stimulates it. Finally, the thyrotropes are the least abundant cell type in the anterior pituitary, comprising just 5% of the gland. Their output, thyroid-stimulating hormone, drives the thyroid gland to produce T3 and T4. These circulating hormones then exert the predominant negative feedback on thyrotropes. If a patient has long-standing untreated hypothyroidism, the absolute absence of negative feedback causes the thyrotrope cells to undergo hyperplasia, leading to physical enlargement of the pituitary gland visible on an MRI. Cellular abundance does not equate to systemic stability. Under pathological conditions, even a 5% cell population can structurally remodel the entire gland. Because the pituitary operates on rapid peptide pulses and feedback loops, static blood draws are often difficult to interpret. You have to capture highly dynamic kinetics in a single moment in time. This 24-hour graph illustrates the volatility of growth hormone. The bursts are so sporadic that random daytime measurements are undetectable in half of all healthy adults. Because random draws are unreliable, checking for GH excess requires a glucose suppression test. A normal pituitary will respond to a glucose load by suppressing GH a low 0.7 micrograms per liter in women. For baseline assessments, we measure IGF-1 instead. Produced by the liver in response to GH, IGF-1 circulates tightly bound to the carrier protein IGF-BP3, creating a stable diagnostic proxy that avoids the problems of pulsatility. This distinction separates absolute growth hormone deficiency from Laurent dwarfism. In Laurent dwarfism, a GH receptor mutation means the pituitary pumps out massive amounts of growth hormone, but the liver never produces IGF-1. Looking at this graph of the ACTH axis, we see a different pattern. ACTH secretion is driven by the amplitude of the pulses, creating a predictable circadian wave that peaks around 6 a.m. Because cortisol follows this same pattern, diagnostic assays for the adrenal axis must be strictly time-gated. If the blood is not drawn during the morning peak, the data cannot be used to accurately assess the axis. By contrast, assessing the thyroid axis requires less temporal precision. TSH has a relatively long circulating half-life and modest pulse amplitudes. Because serum levels remain steady throughout the day, a single-point measurement is sufficient for an accurate diagnosis. Interpreting these axes requires aligning the lab draw with the specific secretion kinetics of the hormone in question, ensuring that a single snapshot in time actually reflects the biological reality of the gland.